You had a question, right?
Yes, doctor.
Go ahead, please.
What does decide the appropriate transport protein?
Hmm. Okay, for every ion, there's a specific transport protein that will have enough, the size, the configuration that will match that ion. So, for example, we have calcium transport proteins, we have sodium transport proteins. It is similar to a channel, but slightly different in that, it opens to one side, takes the ion and lodges it inside the cell. Um, can one transport protein work for more than one ion? Again, yes, within certain limits. Did I answer your question?
Yes, yes.
And you can probably search if you like any extra information. I really would like to see more activity. When I put this, I know it's a short time we are having together, but I think, can you see the screen now?
Yes, we can.
Yes, we.
Yes.
Yeah, you just double click anywhere and you attach whatever you want. So here you can say, okay, attach this. Oh, I opened something. Okay, never mind. So you just double click anywhere, add any file, add any article, anything you would like to, or a question, and we will all help you answer it. I like this link between yourself as colleagues. And this is why I was keen to have the boys and girls together, because the bigger the class, the more interaction and the more ideas we get. Right. So these are the slides you're talking about, right? They're slightly different. I can see that. Because here, lecture 3 starts with the transport. This is the one you took with Dr. Faisal? These are the ones you took with Dr. Faisal?
We haven't taken anything with any other doctor.
Oh, I'm sorry. Why am I confusing? Yeah, sorry. So this is actually, we were in the right slides, so because these are the slides that are yours. Right. Okay. Next week, you will be taking the 5, 6, 7, 8 with Dr. Farwat Jamaluddin. He's been giving these lectures for the last year, so I am the new one here. So we talked about mechanisms by which the cell membrane can allow substances inside the cell. And now we want to talk about transport across the cell in more details. What are the protocols that the cell takes in order to allow? There are simple protocols, which we call as passive transport and active transport. So it's very simple. Passive means there's no energy. Active, there is energy. And because here we are transporting from low to high, then I will need energy. Just like if you are cycling uphill, then you need more energy. Because here we are going from high to low, so I'm actually going by gravity. I don't need energy, so no ATP is required. So this is straightforward, but the only confusion that I see in the exams is when it comes to facilitated diffusion. Here, people confuse it and think because it is facilitated, because there is a carrier molecule, then it is active. But actually, there's a carrier molecule, but it is still passive because there is no ATP hydrolysis or requirement. So we will talk about 3 types of passive transport: simple diffusion, facilitated diffusion, and osmosis. And we will talk about 3 types of active transport. So usually passive transport, as I said, is like going downhill from high to low. And there are 3 types: simple diffusion, passive transport. The first one is simple diffusion, where molecules can directly move through the phospholipids of the plasma membrane. Can you give an example which can go with the direct?
The oxygen.
Gases
Excellent.
in general.
Water.
Water
No,
and oxygen.
Alcohol.
What is water simple? Yes. So it has to be lipid soluble, it has to be small, or gas, as you said, in oxygen. So what is diffusion? Let us define diffusion first. You see those green and yellow completely separated by membrane, and then they mix up, okay? So diffusion is the movement of molecules from high concentration to a low concentration until they are equally distributed. So the drive for the movement is the difference in concentration. Sometimes the drive is the difference in electricity. So either the charge or the concentration will give me a drive to diffuse. And the diffusion rate is related to certain factors: temperature, pressure, state of the matter or the substance itself, size, concentration gradient, and surface area of the membrane. All of these factors affect the diffusion rate. So we are talking about a passive phenomena where a substance moves from higher concentration to a lower concentration through a membrane. And this one does not require a carrier, does not require a channel, but it is affected by temperature, by pressure, by the nature of the substance, by the size and concentration gradient of the substance across the membrane, how much difference there is from outside and inside, and by the surface area of the membrane through which this diffusion is taking place. Okay, so this is the first type, which is passive, does not require ATP. Now, example, oxygen, carbon dioxide, water molecules, and somebody was not happy with that. I agree, because water is polar, is water soluble, so it will not be moving fast. And then lipids in general, steroids, because they are lipid soluble. Lipid soluble molecules, alcohols, very good. Vitamins, some vitamins, not all vitamins. Which vitamins are lipid soluble? Do you know? Let me ask it differently. Which vitamins are water soluble?
I think I don't know, just an information in my head.
Yes, A is what?
As
Water or lipid?
Excellent.
It's lipid soluble. Another is lipid soluble?
C.
Vitamin C, yeah.
No. A, D, E, and K are lipid soluble. So
Water soluble C.
Water soluble
C is water soluble.
Yes, C is water soluble. Good for you. This is why no matter how much vitamin C you take, it cannot be harmful, because you can wash it away in urine. The same thing for vitamin B. You can take a lot of it. It does not harm because it washes away if it is in excess. But the A, the D, the E, and the K are lipid soluble. So you have to be cautious. You cannot take more than you require. Okay. So all of these, they can pass by simple diffusion. Any small non-charged molecule can pass by simple diffusion. We don't need ATP, we don't need any protein. Right. Why is cell diffusion important? Respiration. Now, how do we do respiration? We do a gas-phase exchange through my respiratory system. So I inhale air, my alveoli extract the oxygen from the air and transport it to the blood or vascular system. It goes via the blood to the tissues, and the cells will have that oxygen. I have diffusion at the level of the lung. So here I have an alveolus. On one side, I have air, which is rich in oxygen. On the other side, I have blood, which is rich in carbon dioxide. So I have arterial blood and venous blood. So the venous one, and I'm talking about pulmonary veins, right, are the ones rich in oxygen, and the arterial ones are the ones rich in carbon dioxide, just vice versa, you see? So the pulmonary artery, the one that goes from the right heart, is the one that has carbon dioxide. So it comes to the alveolus, and the diffusion takes place here. This is one alveolus. It's full of oxygen, and it has to, the diffusion takes place with the pulmonary artery. So the pulmonary artery brings the carbon dioxide filled blood from all over the body, and it exchanges here and it goes in the pulmonary vein to the left side of the heart to go to the rest of the body. Okay, what I want you to know is that there is diffusion. If we did not have direct diffusion of these gases, this will affect my respiration. And I have another direct diffusion of gases at the level of the tissue. So when these capillaries reach to the tissue, they will release oxygen, take away carbon dioxide. At the level of the capillaries, all the time we have diffusion with the cells. So nutrients, glucose, oxygen, and all electrolytes go to the cell, and we get rid of the waste products. At the level of the red blood cell, we have diffusion. So the oxygen that's taken at the alveolus will have to diffuse through the cell membrane of the red blood cells to bind to the, where does the oxygen bind to?
Hemoglobin.
Hemoglobin, exactly. So I'm carrying oxygen by two ways. I can dissolve it in the blood, but this is a small percentage. Most of it is bound to hemoglobin inside the red blood cells. So even when it goes through this membrane to the blood, it has also to go through another membrane, which is my red cell membrane, to bind to the hemoglobin. So I have, and we call this the oxygen carrying capacity of the blood. Now, do you know a poison that affects that oxygen carrying capacity? Does anybody remember something that we can do to a person that he will not be able to carry oxygen in his red blood cells?
Anemia, you mean?
That one, you reduce it because you have less hemoglobin. Yes, no, I mean something like a poison where you have hemoglobin, but it cannot carry oxygen.
Maybe nicotine.
CO.
Yes, who said CO?
Honey.
Bravo, Hanin. CO or carbon monoxide. And where do you get carbon monoxide from? Example, just give me an example. You know that the car exhaust is full of carbon monoxide. So sometimes it happens that somebody sleeps in his car, in the garage with his car on, and they get carbon monoxide poisoning. And what does carbon monoxide do is that it will combine with hemoglobin and not allow it to take any more oxygen. So no matter, even if they give them 100% oxygen, the amount of carrying capacity by the red blood cells is less. Okay, we need it also for medication, diffusion for some medication, for some vitamins. Right, so is it clear, diffusion?
Yes, it is.
Straightforward, nothing involved, no ATP, from high to low, as fast, simple, no interference, okay? So we're not talking about the binding here, by the way, I don't want to confuse you. We're not talking about the binding, we're just talking about the diffusion. Binding with the hemoglobin is something different. We're just talking about diffusion between the alveolus and the pulmonary capillary, and then diffusion between the pulmonary capillary and the red blood cells. We are not talking about the binding, that's not the diffusion. Right, so the second passive transport is called facilitated diffusion. This is slightly confusing because now the molecules can go with the aid of a transport protein. And because the protein is involved, because it is facilitated, people might think that it is active. But please remember, facilitated diffusion is a passive phenomenon. We don't require ATP. Here we have a transport protein that makes it easier. And why do we have a transport protein? Because this molecule cannot go by diffusion. I mean, we need some help. So it works like this. This is simple diffusion. This molecules cannot go by simple diffusion. So I have a transport protein in the plasma membrane to allow it to go. This is facilitated diffusion. And this protein does not require ATP. So it's completely passive phenomena. It's from high to low. Okay, so again, the two main rules for passive transport is that it's from high to low. So it's together with concentration gradient. And second is that we don't require ATP. Okay. So here it's high, it acts to the protein and then throws it outside. Okay, from high to low. So what kind of ions move by facilitated diffusion? Through the plasma membrane. Think of it. Sodium, potassium, chloride,
Ions.
sugar, yes, amino acids, water. Water can go by both diffusion and facilitated diffusion. Okay, so all of these ions are actually moving through the membrane by facilitated diffusion, and it includes channels, it includes all kinds of carrier proteins. Right, so this is facilitated diffusion. What kind of protein is this one? Channel protein.
Channel.
Excellent. So a channel protein like sodium channels, potassium channels, chloride channels, they are like this. They are just like a bridge, and they make it easier for these ionic particles to go to the other side. These are called
Carrier.
carrier. Exactly. So they cling and they open to the other side. So one time they open outside, one time they open inside, or vice versa. But it still obeys the rule. From high to low, no ATP. Okay, so it is a passive phenomenon. So just remember, if the ion can go by diffusion, why do we need facilitated? But sometimes we need a faster kind of transport, and this is a more sure than we use this kind of mechanism. But still, it's a passive phenomenon. So remember, glucose, amino acids. I need nutrients for the cell, and they cannot move by diffusion, so I use these kinds of mechanisms. Okay, now because it requires this bridge, then the rate is limited. Just like when you go from here to Bahrain, we cannot go like 1,000 cars at the same time. There is a limit. There is a rate. So what is the limit? The cell can regulate it. So some cells will have more carrier proteins, so these molecules can go faster into them. Some cells will have less carrier proteins, and so is for channels. So I have a control over how much I can have by facilitated diffusion. Do I have a control over diffusion? Simple diffusion? No, because
No,
it
no,
just
you don't.
goes directly. Exactly, it just goes directly through the cell membrane. So this is the advantage of having facilitated diffusion, is that I can control it. These are examples of facilitated diffusion, why it is important. So can obtain food, neurons can communicate, and what do we call the messenger between neurons?
Impulses.
That's the signal, electric signal, but I'm talking about a certain chemical.
Hormones.
We call them neurotransmitters. You will take 8 lectures on the central nervous system, and we'll talk about neurotransmitter, which is a messenger between neurons. In the small intestine, we can transport food, which you have already assimilated and digested into the cells, to the bloodstream, to make use of it. In the muscles, we can allow muscles to contract by this kind of facilitated diffusion. Right, so 2 types so far: simple diffusion, facilitated diffusion. The third type, which is also passive, is called osmosis. And here, we are not talking about the molecule, we are talking about the water.
Yes.
So please be clear about it, because otherwise you'll be totally confused. When we were talking about high to low in the previous 2, we were talking about the solute or the ion or the substance. Here we are talking about the water. So these are water molecules, right? It's not a very good picture, I'm sorry. But, okay, let me see if I have a better picture. Okay, let's read the definition and then we will look for the picture. So osmosis is the diffusion of water through a semi-permeable membrane, which is my plasma membrane. And It is the water which is moving from the high water to low water. Concentrated, I mean, lower concentration of water. So it is still high to low. Still, we don't use ATP. So again, it's a passive phenomenon. Now, osmosis is extremely important. Why? When I want to treat a patient and I want to put an IV solution, I have to use an isotonic solution, meaning I will use the same osmolarity of the IV fluid to the osmolarity of blood. If I use a different osmolarity, what happens? This is the picture I was looking at. I was looking for. So here there is more water, less solute. Here there is more concentrated salt. So the water will move from here to here. You can see the water moved from here to this side. So this one become higher. Why? Because this is the concentrated side. So it will shift or will suck the water, if you like, by osmosis. We call it the osmotic pressure. Okay. Now, why osmosis important? Cells remove water by cell respiration. So during the respiration, the cells, they remove the water. Large intestinal cells transport water to bloodstream. So when you drink water, the large intestine will take most of that water and take it to the bloodstream by osmosis, because the blood is more concentrated than your gut. And the kidney will take the water from the urine in the distal and collecting duct by, again, osmosis. So all of these osmotic pressure gradient help me to conserve water in a passive phenomena without requiring ATP. Okay, let us see what happens when you have some cell in a solution. My red blood cells have, we call them, they have 330 milliosmoles. So it's like, which we consider as isoosmotic. So any fluid I put in the blood has to be 330 milliosmoles, so that the fluid will not shift inside and outside the red blood cell and will give me, so I get the medication without affecting my red blood cell function. If by any chance I do a mistake and I give a hypertonic solution, then that cell will shrink because all the water in that cell will be taken out. I have a better slide. Yes, this is what happened to the red blood cell. You know what happened here? We injected a hypertonic solution or hyperosmolar solution by intravenous fluids. And now what happened? The red blood cell, instead of being nice and disc-shaped, which is optimal for oxygen transport, is horny like this and can easily break, because all the fluid inside the cell left by osmotic pressure to the blood. So we need to have isotonic, anything we put into the body has to be isotonic, same as the blood or extracellular fluid, so that we don't create those shifts in fluid and affect my cellular function. I like this slide because if you put hypertonic solution in blood, this is what happened to the cell. They will shrink. The water will go out from the red blood cells and the cell will shrink. If you put isotonic, the cell will be happy, normal, biconcave disc, and it will carry oxygen in an optimal way. If I put hypoosmolar or hypotonic, then the opposite will happen. All the fluid will go inside the red blood cell, and you see what will happen? The cell will become, this concavity will disappear, the cell becomes a sphere, and it's very easy to rupture. And most of it actually will rupture and cause hemolysis. And you don't have any oxygen carrying capacity then. Okay, so you need to have this situation B, where everything is isoosmolar or isotonic. Now, these conditions also occur in cases of, let's say, heat stroke, when you have lost a lot of fluid and there's hemoconcentration, or in cases of diabetes insipidus, where somebody is drinking a lot of water because of psychogenic or because of um, a lot of ADH, inappropriate secretion of ADH. So they have a lot of water in their body, their blood is diluted, and their blood cells look like this. Do you know of any condition that looks like this, that's hereditary? And it's not uncommon. Do you hear me, guys?
Yes, doctor, we hear you.
Yes, yes, we can. Yes, yes.
Okay. Do you know of any condition which is hereditary that will make your red blood cells look like this? It's familiar.
No.
Okay.
Hypertension.
I think white blood cells.
No, we're talking about red blood cells. It's called spherocytosis. So these people, it goes in families. Instead of having biconcave disc red blood cells, they have spheres. And their cells are very likely to rupture anytime. They're fragile. So they have a lot of hemolysis. And these people, they end up having anemia. So one of the causes of anemia is spherocytosis. But we're not talking about that. We're talking about a condition where there's dilution of the blood or excessive fluid leading to swelling of the red blood cells, and this affects their function.
I think
so osmolarity. Yes. So this is hypertonic, this is isotonic, this is hypotonic or osmolar. Where they say tonic or osmolar is the same thing. This is a different picture. So when I have hypertonic solutions, the fluid leaves the red blood cells and they become just like in the sickle cells. Sometimes they look not regular. They cannot. Now, why do we need them to be in this shape? Because this shape is optimal for oxygen transport and optimal for traversing the small capillaries without rupturing. When it's ragged and it has edges like this, then when it goes through the capillaries, it will be hemolyzed. This is what happens. The cells are happy when there is isotonic solution put to them. This is what happens when there is hypotonic solution. So the water goes into the cell, they swell, and they rupture easily. Now we finished the three types of passive transport: simple diffusion, facilitated diffusion, and osmolarity or osmotic shift. We come to the active. So when we talk about active, we are talking about a transport which requires ATP or energy and which can go against the gradient. So it can go from low to high. And this is a very important mechanism of transport. One of the most important one is the sodium potassium ATPase pump. Have you heard about that pump?
Yes. Yes.
Excellent. So I have a pump in my cell membrane that all the time brings 3 sodium outside, 2 potassium inside. In every cycle, it throws sodium outside, brings potassium inside. And this is a very important pump. I mean, it requires ATP for each cycle. And it's important to put back the ions in the right place after an action potential. So when the cell signals, sodium goes inside the cell. And then when we return the potential to normal, potassium goes outside. So we don't want that. We want them to go back to their normal concentration. So this pump with every turn is putting sodium back outside, putting potassium back inside, and so on. But it requires ATP because it is going against electrical gradient. So what do we do? This is sodium after the potential. Normally we said sodium is outside, right? And potassium is inside. But after the signal, because we open the sodium channels and we allow them to go in, and then we open the potassium channels and we allow the potassium to go out. Now, at the end of the potential, this will be reversed. So I don't want that. So the sodium ATP will take 2 ATPs to put the sodium outside, potassium inside. So I need 2 turns, 3 sodium out, 2 potassium in, 3 sodium out, 2 potassium in. It's an active phenomenon. It goes against electric or chemical gradient, and it requires ATP. So whenever I need to move from low to high, I will require energy. Okay, and I might use proteins which we call as pumps, like the sodium potassium ATPase pump. So don't confuse. So we said in the passive, we have simple diffusion, and we have facilitated diffusion. Most of these are passive, they don't require ATP. Here, I'm going against gradient from low to high, and then I will need energy, so I will need ATP. So this one is active. If the transport itself requires the energy, we call it primary active transport, like the sodium potassium ATPase pump, meaning that the pump itself is taking the hydrolyzing the ATP to produce the movement. So why do we use my energy? Energy is like currency, it's like money. Why do I pay for this kind of transport? It has to be something important. I will not pay for something that is not essential for the cell. So most of the things that go by active transport are ions, amino acids, glucose, some nucleotides, so some essential material for the cell function, or some harmful material that I want to actually get out. So I do that by active transport. So I'm spending energy, I'm spending money, or to maintain internal condition, regulate volume of the cell, control pH, or re-establish normal concentrations like the sodium potassium ATPase pump. All of these conditions are reasons for which we have this active transport. So in the sodium potassium ATPase pump, which is primary active, we have 3 sodium out, 2 potassium in. Please remember, we are throwing sodium out, putting potassium back. But most of the sodium is already outside, so I'm going from low to high. And most of the potassium is already inside, so I'm going also from low to high. So in either condition, I'm going against concentration gradient. And we split the ATP to ADP, and we produce energy. Okay. So I'm bringing sodium from here to here, from low to high. I'm bringing potassium from here to here, again from low to high. Because I'm going against gradient, then I have to spend energy. Now the secondary active transport is like somebody who's depending on something else. So when I transport sodium and amino acids, they actually go together. So let me just play this back. It doesn't want to play back. Yeah, do you see it now? Sodium
Yes.
in, amino acid out, and they go with the same calories. So when we are talking about ATP, the sodium potassium ATPase, you remember? Which brings sodium in, it brings sodium out, potassium in. So together with the sodium, now this is different. This is bringing sodium in and bringing the amino acid out, right? So it's linked, we are linking two, we call them antiport. We are linking two substances together in the same carrier molecule. We call this counter transport. And this is a very important principle in the gut and in the kidney. Okay? Now, the transport of substances at the same time in opposite direction without ATP. So why do we call it active if we are not using ATP?
Because it's from low to high.
Maybe it uses energy, but not ATP from the same molecules that get transported.
Excellent suggestions, but the answer is in the previous slide. You remember the sodium
we
potassium?
used it in the primary stage.
Yes. Who said that?
Me, Sajjad.
Sajjad, bravo عليك. Then, although we don't use ATP by this particular protein, it is dependent on the gradient created by the sodium potassium ATPase pump. So we call them secondary active. Let me give you an example. Let's say that I am paying somebody to cook for your class, cook the lunch or breakfast or whatever. And I give you that lunch, breakfast free. So you're actually eating the lunch free, but somebody else is paying for that person who's preparing the lunch. So it's not a free service totally, but for you, you consider it free. So this is exactly what's happening here. These molecules are getting transported without ATP being spent by this protein. But the gradient created by the sodium is produced by the sodium potassium. So this depends on the primary. But it itself, it does not require energy. Okay, so the transport of two substances at the same time in opposite direction is called antiport. We don't require ATP, but it depends on the ATP produced by the primary active transport. There will be a better slide, and I'll give you also a question maybe to answer. So I have counter transport, I have co-transport, where both are going in the same direction, like this one. Sodium and glucose, the very good example. In the gut, when we take the food, the sodium and glucose are being absorbed together. We call this co-transport because they are going in the same direction. This one was counter transport, they're going in opposite direction. Or gated channels. So I have a channel that allows molecules, and it can close or it can open. Okay, mind you, we are talking about active transport. So even if it doesn't require ATP by itself, it is dependent on some primary active transport. Right, I will explain the primary and secondary in a minute, but let us go to the bulk transport, which is very simple. Let's get it out of the way. Here we are transporting large molecules like a big virus or a big bacteria or big, let's say we want one organelle is hydrolyzed, we want to get rid of it. So this is inside the cell, this is outside the cell. So we digest it, we make it small, and then we just fuse this vesicle with the cell membrane, get it out. We call this exocytosis. Okay, and usually these are bigger substances than the things that we can diffuse. So there are three types, phagocytosis, like the ones that the macrophages do. They produce pseudopods, they engulf the material and get it inside them. Pinocytosis, where it is just for water or droplets, or receptor-mediated endocytosis. So there is recognition, like the antibody-antigen complex. So this is an example of, we are engulfing, we're making pseudopods, engulfing a bacteria by macrophage. So this is phagocytosis. It takes it in, and here it can play with it and put some digestive material and kill it. Will not allow it to harm the body. Okay, so this is an example of, so now the lysosome is putting the enzymes to digest and kill this bacteria. This is one of the immune mechanisms. So endocytosis, phagocytosis, sorry. And phagocytosis is an example of endocytosis. Exocytosis, you have some, is important in neurotransmitter and neural communication. There is a neurotransmitter synthesized within the vesicle. It comes, it docks with the membrane and opens up, and it is released to act on a receptor on the next neuron. This is exocytosis. Phagocytosis again can be called as cell eating. So the cell eats the bacteria. Pinocytosis can be called as cell drinking. It takes droplets faster than simple diffusion or facilitated diffusion. When the intestinal cells, kidney cells take water. He said all of this. Okay, so let me look at, why don't I have the activity? Should be having an activity here. Okay, so this one, can you see the slides?
Yes.
Yes, we can.
Yes.
Right.
Yes, doctor.
So what is this one? The one on the left. Type of transport.
Sodium potassium bomb.
Excellent. So what type of transport is it? It's written there. It's primary.
Primary active transport.
Yes, the counter transport is secondary. Now we're talking about the primary active transport because of this one. The clue here is this one, because this pump will not act unless I have ATP. So in a person who's dead, who cannot produce energy or respire or produce ATP, you will not have this pump acting. There is direct dependence of this pump on ATP. So it is a primary active transport. It's not a co-transport. Although they are going in the opposite direction, the co-transport is a secondary mechanism. Okay, this one, there's no ATP. Do you see ATP anywhere? No. But I'm depending on the presence of sodium, which is created by the ATPase pump. You see, this is why I call it secondary active transport. So here, sodium is going in and this substance is going out. So it is, so they're going together, sorry. So this is co-transport, but we don't use ATP by this protein, but the presence of sodium is created, the presence of sodium is created by this primary mechanism. So we call this a secondary active transport because it depends on the ATP that has been used by another pump. We have the sodium glucose co-transport is a very common example, important example. That sodium and, so you see when you have low sodium, what happens? You cannot absorb glucose properly. If a person has very low sodium, hyponatremia. Okay, is it clear now the difference between primary and secondary?
Yes.
Both
Yes.
are considered active. We cannot call it passive. Okay, still it is from low to high, and this is again is an active phenomenon. Good, I'm glad. Again, another way, primary active transport. There is ATP here involved directly, like the sodium potassium ATPase pump, sodium outside, potassium inside. This is primary active transport. Here, I have co-transport of glucose and sodium. It uses the same sodium that was brought out by the primary. So it does not hydrolyze ATP, but it is depending on the sodium produced by the ATP. So it is a secondary active, and this is called we can either call it co-transport or symport, meaning they are going in the same direction. If they're going opposite direction, we call them co-transport. Can we just, for the sake of fun, do this exercise?
Okay.
Excuse me, doctor.
Yes, please.
You mentioned that if it goes opposite directions, it's co-transport.
Only if it is secondary.
Oh, okay.
Yeah, meaning, okay, it is a co-transport, but the issue is, if it is primary active transport, then it is like the sodium potassium ATP pump. We don't want to confuse with the secondary. So the secondary transport, we call it either co-transport or counter-transport or semi-port or counterport. You see, they're the same thing. Semi means in the same direction, like co. Counter-transport means in opposite direction. So most of you think it is number 2, right? Can you see the slides now when I put them? Do you see the answers? Do you see the responses of the group?
No, no.
Okay. So let me stop share and share this one again. So I thought I was sharing with you. Do you see it now?
Yes, we can.
Okay.
Yeah, yes.
So most of you have chosen number 2, which is correct. Okay, because facilitated diffusion is a passive phenomena. We don't have ATP, and here in number 1 you have ATP clearly there, so it cannot be facilitated diffusion. Right? Can we do? Let me see the next slide. Okay, can we answer this one now? Do you see the next question?
Yes, we can.
Yes, we can.